Okay, so welcome to this next video in which we are discussing the hemostasis pathway. Okay, so we're trying to see how, if we've made a cut in the wall of our arteriole, how do you form a hemostatic plug which blocks blood from leaving the lumen of that arteriole and going into the extracellular fluid. Right, okay, so we've discussed how you can get platelets starting to adhere to the area of damage, and they're adhering via an interaction between the glycoprotein 1b95 on their surface membranes and a von Willebrand factor protein, which is on the surface of the damaged endothelial cells, because it was on the basolateral side, but now it's been exposed, basically, to the platelets. Okay, so what happens next is that you need platelet activation, okay, and the way that you get this is through platelets being exposed to proteins that they would not usually be exposed to. So remember, platelets are usually within the lumen of the blood vessel, so usually all they see is the other constituents of the blood and the apical surface of the endothelial cells. That's all they see. They do not see a whole host of proteins. Now, one of the proteins that they would not ever see in the lumen of the blood vessels is collagen. Okay, now collagen is the main, one of the main proteins that makes up the basement membrane here. It's also one of the main proteins that makes up all connective tissue. So basically, underneath the basement membrane, you have another layer of connective tissue known as the subendothelial space. That will be full of collagen. I didn't show that on my diagram when I was just drawing the blood vessel here because I wanted this to be a nice simple diagram. But underneath the basement membrane, before you go to tunica media, what you have is a little strip known as the subendothelial space. So I'll just discuss this a little bit now. Okay, so just to expand on this diagram of the structure of the blood vessel, what you initially have is here is the endothelial cell. Then you have the basement membrane on which the endothelial cell is sitting here. Okay, so I'll draw the nucleus of the endothelial cell to make it look more convincing, and I'll even label it up. So this is an endothelial cell, an EC. This is the basement membrane, okay, basement membrane, and then underneath the basement membrane you have a portion of connective tissue known as the subendothelial space, or the subendothelial connective tissue. So what colour should I draw this in? I'll draw this in red, and it's going to be thicker than the basement membrane, so I'll make it like this. Okay, so this is a region made up of mainly collagen, okay, so this is the subendothelial connective tissue, or the subendothelial space. Subendothelial space. And then finally, underneath that, what you have is a very thin layer known as the internal elastic lamina. And this blue highlighter has got extremely dirty, hasn't it? Okay, so this is the internal elastic lamina, and then outside of that you then have the smooth muscle cell there, where you have where you have the smooth muscles, and that's called tunica media. So internal elastic lamina. Okay, so all of these are actually the tunica intima. So this whole thing now is the tunica intima. Before, I simplified it down by just talking about these two layers, but this is really the full structure of the tunica intima. And then outside of that, you then have the tunica media, which is this layer of smooth muscle cells. Okay, so the subendothelial space we've discussed is made up of mainly collagen. The internal elastic lamina is made up of the protein elastin, a very elastic protein. Okay, right. So the point is that when you cut through here, okay, what's going to happen is that these platelets are also going to be exposed to all of these proteins. In particular, one of these proteins is collagen, basically. Okay, now collagen has a fantastic way of activating platelets. So when platelets meet collagen, so if you've got blood coming out here, what's going to happen is that platelets are going to meet the collagen, and this exposure to collagen is actually going to activate them. Now, you might think, well, is this platelet actually going to be able to see the collagen when it's adhered to the endothelial cell? And the question is, well, maybe it might be able to. It might just manage to see some collagen and therefore become activated. But what we're going to see in a moment is that there is a chain reaction, basically, where one platelet becoming activated sets off another one to become activated, which then sets off another one to become activated. So even if this one which has actually adhered to the von Willebrand factor cannot actually see the collagen itself, a friend basically might come through this hole and get to the collagen, and it might undergo the activation process and will therefore trigger, through this chain reaction, this one which is adhered to undergo the activation process. Okay, right, so let's discuss the activation process of a platelet. So let's imagine that our platelet which has undergone the adhesion actually is going to see some collagen and is going to undergo activation. Okay, so what happens when a platelet undergoes activation? And I'll just write somewhere down platelet activation. Okay, so we've had platelet adhesion so far. All this von Willebrand factor GP1b95 does is it holds the platelet near the area where you've had this terrible disturbance. Okay, for further things to happen you need platelet activation, and this is produced by exposure to collagen. It's also produced by exposure to something known as tissue factor, or also called factor III, which is a protein on the surface of cells, basically. So all of the cells which are not endothelial cells, well, at least are not apical surfaces of endothelial cells, have this protein known as tissue factor on their surface. It's also called factor III, and we'll be seeing this later. It's responsible for the extrinsic coagulation pathway. Okay, so basically, if a platelet was to go out far enough, it would meet cells, okay? You know, cells which made up the blood vessel, or even maybe it could go out into the extracellular space, and then it would find cells which were not endothelial cells. So basically, by meeting other cell membranes that are not just the apical membrane of endothelial cells, you will meet tissue factor, and that will activate the platelets as well. So what happens in platelet activation? Well, two things. The platelet, well, there are more than two things, but we're going to concentrate on two main things. Basically, when a platelet is activated, so let's have a platelet here. When a platelet is activated, what happens is it releases two major things which we're going to study at least. So one of them is it releases thromboxane A2, which is often abbreviated to TXA2. So the full name for this is thromboxane A2. Okay, so thromboxane A2. And the other thing that these platelets release is ADP, adenosine diphosphate. Okay, now, so when this platelet undergoes activation, when it sees collagen, basically, or tissue factor, it's going to activate and it's going to start releasing ADP and thromboxane A2. Now, we'll discuss what thromboxane A2 is going to do in a moment. We'll discuss the chain reaction first. So, the chain reaction is that once one platelet has found some collagen or tissue factor and has undergone this activation process, the release of ADP is then going to go and activate more platelets. So platelets basically have a protein on their surface here, okay, which is an ADP receptor. So this is an ADP receptor. Okay, so ADP will bind to this ADP receptor, and when it does, it causes this second platelet over here to undergo activation. So I'll highlight this ADP receptor in pink here. Okay, and this highlight has gone horrible dirty colour as well. I need to clean them. Right, okay, so we've got this ADP receptor here, and when ADP binds to that ADP receptor, it's going to cause the ADP receptor to become active, and it will then lead to downstream pathways which activate the platelet. So this platelet will undergo the same process, it will start dumping out ADP and also thromboxane A2. Now, so ADP is involved in this chain reaction, basically. It's involved in one platelet causing another platelet to become active, and then this will go off and cause more platelets to become active. This is involved in the amplification of platelet activation, basically. However, what's the point of this activation? Well, that must mean that this thromboxane A2 is somehow very, very important. And we'll discuss in a moment why it's so important after I just give you a little pharmacological correlate. Okay, so there's quite a famous drug, a very widely prescribed drug to protect people against diseases involving thrombosis. It's an antithrombotic drug, so it's given to people who are at risk of having stroke or MIs or things like that. Okay, and this drug works by binding to the ADP receptor. Okay, and this drug is known, its name is Clopidogrel. So if you go into Boots and you go to the pharmacy counter, then generally if you look for long enough, if the queue is long enough and you have a look at the drugs behind the counter, you will probably see Clopidogrel somewhere. It's a very widely prescribed drug now. Okay, and the way it works is that it binds to this ADP receptor, basically, and stops the chain reaction being set up, because if the platelets have Clopidogrel bound to them, then the ADP cannot bind to the ADP receptor, so you can't get this chain reaction of activation of platelets. So the number of platelets which become active is hugely reduced, because it's only now those that actually were directly activated by the collagen or tissue factor. So remember, there's two things that can activate this: collagen and factor 3. I'll just denote it as factor 3. Okay, right. So that's an interesting correlate, and this drug is stops your blood from clotting. Basically, it stops you from being able to form platelet plugs. Okay, so it's quite bad if you bleed whilst on this drug. Well, if you cut yourself on this drug, your blood will take a very, very long time to undergo hemostasis. Okay, but it will protect you against thrombosis, which is basically where you get the hemostasis process activated, but there's no need for it to be activated. So in physiology, basically, if you cut your blood vessel, what should happen is you should activate the pathway of hemostasis and form a hemostatic plug in the wall of the artery, or the arteriole. Hopefully you haven't cut open your artery. So in the wall of your arteriole, and stop blood coming out of that arteriole. Whereas thrombosis is when you get this entire pathway activated, but it just activates in a blood vessel which has not got any sort of a cut in it. So you get you get hemostasis, all the hemostasis pathway activated. You form these sort of plugs in the bloodstream, and these are very, very dangerous because they can block blood vessels and stop blood getting to places. And if you get these thromboses in blood vessels supplying portions of the brain or the heart, that's very, very dangerous, and it can lead to stroke if it happens in the brain, because portions of the brain can die if you don't supply blood to them, and it can cause heart attack if it happens in the heart, because again, the heart muscle dies if it isn't receiving oxygen. Okay, so if you want to stop thrombosis from occurring, if you want to stop the formation of these plugs just in the middle of blood vessels, then Clopidogrel is a good drug to use. Okay, right, but that's just a correlate. We'll have a whole video on antithrombotic drugs, and we'll talk about things like warfarin and whatnot. But just to add that in because it seems appropriate. So back to the process of hemostasis in health. What should happen is that a platelet should be activated by exposure to either collagen or tissue factor, and this will lead it to release ADP, adenosine diphosphate, and thromboxane A2 here, and the ADP will then cause this chain reaction of more platelets being activated. Okay, now, there's no point in the platelets being activated unless it's actually going to do something. So that must mean that the release of thromboxane A2 is somehow very important, and it is. So firstly, what thromboxane A2 does is it causes vasoconstriction. Okay, so it's going to activate vasoconstriction of the blood vessel. So you're going to basically, if we draw our whole blood vessel here, so here's our blood vessel with this cut down the middle here. Then what's going to happen is you're going to get thromboxane A2 being produced all over the place here. So if I draw the thromboxane A2, here is loads and loads of thromboxane A2 being produced in the vicinity of this cut. Okay, and what it's going to lead to is it's going to lead to the contraction of the smooth muscle cells around around the blood vessel, that line the blood vessel, that are within the tunica media, and that's going to lead to the blood vessel shrinking, basically. So what's going to happen is you're going to get a blood vessel that looks maybe instead like this. So the lumen of the blood vessel is going to get hugely reduced, and the aim of this is to stop more blood coming into this portion of the vascular system where you have a cut. So it's to stop more blood being lost from this cut. Okay, so vasoconstriction, and it leads to the hemorrhage going down. So this is actually a very, very important part of hemostasis, because remember, the definition of hemostasis is not the formation of a hemostatic plug in the place where you have the cut. The definition of hemostasis is the prevention of hemorrhage, basically, to reduce the loss of blood from the cut. Okay, so vasoconstriction is part of that, to reduce the blood that's getting to this portion of the blood vessel where the cut is present, and therefore reduce the loss of blood through the cut. So that's one of the functions of thromboxane A2. It's also essential in actually starting the production of this hemostatic plug. Okay, now to understand how it does this, we need to we need to introduce another key player that is on the membrane of platelets. So basically, another protein that is on the membrane of platelets, okay? So let's draw another platelet here. And we're now introducing another protein, and it's another glycoprotein, so it's got the title GP, okay? And I will highlight this one in green. Okay, and this protein in green that's on the cell surface membrane of our platelets is a protein known as glycoprotein, or GP2B, so Roman numerals again, 2B, and then they put a / 2A. So it's a protein complex, basically. Okay, and this protein is going to bind to thromboxane A2, or thromboxane A2 is going to bind to this protein. So let's have thromboxane A2 coming in here. So here comes thromboxane A2, and we'll have thromboxane A2 in vivid purple here. So in comes thromboxane A2, and what it's going to do is it's going to activate this glycoprotein 2B/2A. So this is thromboxane A2 coming in here. And when the glycoprotein 2B/2A becomes activated, what now can happen is it can bind to fibrinogen, and we'll continue this discussion in the next video.